इन स्लाइड्स में हम बात करेंगे डीएनए स्ट्रक्चर के बारे में. Watson, Crick and Wilkins got the Nobel Prize for the development of the DNA structure model in 1953. And due to this landmark contribution towards the science, they got this Nobel Prize. So, what is the DNA structure? How it looks like? The three-dimensional structure derived from Watson and Crick model is composed of two side-by-side strands or chains of nucleotide which are twisted into a shape of double helical structure. The two nucleotide strands are held together by hydrogen bonds between the bases of each strand, forming a structure like a spiral staircase. So that looks like a spiral staircase, the steps of which are made up of these nitrogenous bases, and the backbone of this is made up of sugar and phosphate group. The steps contain the base pairing of the nitrogenous bases, and that determines the complementarity rule. और ये complementarity rule जो है इसके तहत सारी की सारी information जो है वो transfer होती है, whether that is in the form of formation of new DNA molecule from the parent molecule, from that the formation of messenger RNA molecule from DNA, or the formation of protein from the messenger RNA molecule. So all of these require this complementarity base pairing, जिसको हम complementarity rule भी कहते हैं. So what is this complementarity rule? A always binds with the T, and that makes two hydrogen bonds, and G always binds with the C, and there are the formation of C hydrogen bonds between C and G. It means A always code for T in the DNA, or U in the RNA, and G always code for C. The backbone of each strand is formed of alternating phosphate and deoxyribose sugar units that are connected by phosphodiester linkages or phosphodiester bonds. So here you can see these phosphodiester bonds between the sugar and the phosphate group. So phosphodiester bond is a group of strong covalent bonds between a phosphate group and two 5-carbon ring carbohydrates, pentose sugars, over two ester bonds. Sugar phosphate backbone is said to have a 5' to 3' polarity or direction. The double-stranded DNA molecule, the two backbones are in opposite direction. So this is why we say that both strands are in opposite direction. One is 5' and to 3', and that is also called the positive strand or sense strand. The other strand is from 3' and to 5', and that is called negative strand or antisense strand. Each base is attached to the one carbon atom of a deoxy sugar in the backbone of each strand, and faces inward toward the base on the other strand. Similarly, the base pairs, which are flat planar structures, stack on top of one another at the center of the double helical structure. A single strand of nucleotide has no helical structure because that is the property of the double strand. The helical shape of DNA depends entirely on the pairing and stacking of the nitrogenous bases in the antiparallel strand. Next is the forms of DNA. There are different forms of DNA, as you can see. aDNA, bDNA and zDNA. So there are three forms of DNA. bDNA is the most common form that is present in the cells, that is normally present in the cells. And this form is right-handed, twists clockwise, 10 base pair per turn, 34 angstroms or nanometers per complete turn. Minor groove is narrow and major groove is wide. So here we will discuss in the next slide about these minor and major grooves. In a solution which is higher in salt concentration or with alcohol added, the DNA structure may change to an A form, which is still right-handed, but every 2.3 nanometer makes a turn. So that is kind of shrinked DNA, the A form. There are more base pairs per turn. As in case of bDNA, there are 10 base pairs per turn, but in case of A form of DNA, there are 11 or more base pairs per turn. So A form, that is conformation, is favored when DNA is dehydrated. As I told, when we apply high salt concentration and alcohol, high osmotic pressure develops, and that leads to dehydration. And this DNA is wider and flatter than the bDNA. That is also right-handed DNA. 11.6 base pairs per turn, 34 nanometer angstroms per complete turn, deep major grooves and shallow minor grooves. The zDNA, because its backbone forms a zigzag structure. Here you can see the zigzag structure. The zDNA conformation is favored in certain high GC content sequences or GC rich sequences. Both A and B DNA are right-handed, but the zDNA is left-handed, and that twists anti-clockwise. There are more base pairs, 12 base pairs per turn. That is kind of larger grooves, 44 nanometers per complete turn. A deep minor groove. There is no apparent major groove seen. Reversible change from bDNA to zDNA can take place. For example, zDNA exists under high salt conditions or upon methylation. So this zDNA has a very putative role in the gene expression control or the regulation of gene expression. So this was all about the structure of DNA and different forms of DNA.